All right. Hi, I'm Shingo Kajimura from Beth Israel Deaconess Medical Center and Harvard University. First of all, thank you for watching me, and also for the opportunity to talk about our research. So today, I'm going to talk to you about bioenergetics, why bioenergetics is important for metabolic health, with the emphasis on brown fat. We have amazing capacity to adapt ourselves to a variety of environmental changes, such as nutrition and temperature, exercise, and other stress. On the other hand, a maladaptation to those we call metabolic stress leads to obesity, diabetes, and cardiovascular diseases. So my lab aims to figure out what is really going on during the metabolic stress or adaptation or maladaptation at the molecular or cellular or systemic levels. And the way I look at it is that if we really understand what's going on at the molecular levels, we should be able to rewire the circuits and restore metabolism to a healthy state. So our approach is to study the bioenergetics of brown fat. So first, I'm going to talk about this or overview, what about brown and beige adipocytes. Then I'm going to talk to you about how enhanced, we call browning or beige fat, improves metabolic health at the molecular level. And lastly, I'm going to touch upon the link to diabetes with the emphasis on the role of brown and beige fat in amino acid metabolism. So for the last 10 years, my lab is trying to gain fundamental understandings of brown adipocytes, which is really a great model to study mitochondrial biology because it contains mitochondria at the highest density among mammalian cells. Also, brown fat has high fuel oxidation and dissipate energy in the form of heat. It's called thermogenesis. And these features are completely opposite from white adipocytes, which has a low mitochondria and very low fuel oxidation, and essentially work as energy storage. The best well-known function of brown fat is called non-shivering thermogenesis for body temperature regulation in hibernating animals or infants who don't have enough muscle to shiver. And these brown adipose tissue exist in the intrascapular region, back of the shoulder, and this is really the sort of the well-known biology for brown fat. But for the last 10 years, several major findings transformed this field. One of them is the importance in human health. Adult humans have active brown adipose tissues in these distinct locations from infants, and these are detected by FDG PET scan. Now, many studies, including this large cohort study, show that people with active brown adipose tissue have a significantly lower odds ratio of type 2 diabetes by 50%, and dyslipidemia and many heart diseases. Notably, these effects are independent of body weight because the effect is still significant even when compared with body weight match controls or groups. Another breakthrough is a characterization of the what we call inducible form of brown fat called beige adipocytes or bright cells. In adults, a subset of white adipocytes can adapt a brown fat phenotype known as the browning or beiging of white fat after cold acclimation. And we call them, beige or beige adipocytes. And these are really, in terms of the function, beige adipocytes are similar to brown adipocytes in terms of the high mitochondrial density, oxidation, and thermogenesis. But they are different cell type and in terms of the cellular lineage. And then they're not from, we call skeletal muscle lineage, and they are highly inducible. This is one of the very important feature of beige adipocytes. And then the function and the mechanisms are quite different from embryonic brown adipocytes, and which I'm going to talk more about in a moment. I want to emphasize that humans have beige fat. So my lab cloned the first thermogenic adipocytes in adult humans several years ago. This is a transcriptional analysis of the isolated human thermogenic adipocytes as opposed to mouse beige or embryonic brown adipocytes or white adipocytes. These are principal component analysis. As you can see here, this transcriptional analysis show that cloned thermogenic cells have the characteristics of mouse or the beige adipocytes in mice. So this study, together with other works by other groups, suggests that the browning can be induced in adult humans, and that's very exciting. In fact, a recent study showed that repeated activation of beta-adrenergic receptor or a chronic cold acclimation in people who did not have brown adipose tissue before the treatment actually regain new brown adipose tissue after this chronic treatment or chronic cold exposure. And this recruitment of new brown adipose tissue, we call beiging, is accompanied by improved insulin sensitivity. So these studies really provide compelling evidence that the activated brown adipose tissue, or we call beiging, is associated with metabolic protection. But the question is how, right? So it really why brown beige adipocytes are protective against metabolic diseases such as lipid improvement, lipid homeostasis, glucose intolerance, and insulin sensitivity. And then really specifically is this metabolic benefit of brown or beige adipocytes entirely by enhancing thermogenesis. So this is something I'm going to talk to you today. So so this is the sort of the model, conventional model where brown beige fat makes heat by and then this increased energy expenditure, this leads to body weight loss and improved metabolic health. Is it really model true? So before, what I haven't told you, and I'm sure everyone in this audience know this long-standing notion, is that brown fat or beige fat generate heat by a mitochondrial proton carrier called uncoupling protein 1, or we call UCP1. So this is uncouples proton gradient from ATP synthesis. And UCP1 was sort of thought to be the only thermogenic protein mammals for the last probably 30, 40 years. And then therefore, a field assumed that all the metabolic benefit of brown and beige fat depends on UCP1. Okay, but however though, there's a paradox in the field. I want to show you first example. Here, from evolutional standpoint, actually many mammals don't have functional brown UCP1 protein. One of the very well-known examples is pigs versus wild boar, where pigs lost functional UCP1 gene, and then through that, actually humans picked UCP1 null wild boar as a sort of for domestication. It makes sense because it's really tasty. But instead of making heat, they actually accumulate subcutaneous adipose tissue as an insulator. So the pigs accumulate really thick subcutaneous adipose tissue, and that's why bacon is tasty. But however, though, the UCP1 knockout wild boar, this pig, they're not hypothermia and they're not diabetic. So there must be something that can compensate for the loss of UCP1. Another example from earlier work by Leslie Kozak and Sven Ellenbach work is that this paper showed that UCP1 knockout mouse are not obese or not insulin resistant at room temperature. Okay, so however, works by Brad Lowell and Jeff Flyer's work show that diphtheria toxin mediated depletion of brown fat cause obesity at even at room temperature. Again, there's another example showing that brown fat deficient mice are quite different from UCP1 knockout mouse. Let me tell you our story in our end. So back in the old days when Patrick Seal and I were a postdoctoral fellow in Bruce Bigelman's lab, we worked on the transcriptional regulation of brown fat development, and it characterized this PRDM16 transcription complex, which is required for brown and beige adipocyte development. So this is composed of PRDM16 and EHMT1 and PRDM3. later, my lab really showed that this EHMT1 is the enzymatic engine of the PRM16 complex. And then, so depletion or deletion of this gene, enzymatic complex EHMT1 in brown fat progenitor cells leads to a really striking phenotype. This is the data. So the EHMT1 knockout mouse in a MIF5 specific manner, these mice have sort of really small but presumptive embryonic brown adipose tissue that express many of the muscle genes, but no thermogenic genes are expressed in this presumptive brown adipose tissue. So essentially, this is a brown fat deficient mouse. And this mice phenocopied the double knockout of PRM16 and PRM3 because they physically interact and make a complex. Notably, this gene EHMT1 is deleted, one of the rare human syndrome. It's called Clifton syndrome, and where 44% of the child develop obesity. And this gene is highly expressed in human brown fat. So we deleted EHMT1 specifically in the fat cells, and then the mice who lost EHMT1 only in the fat cells gained more weight, y-axis body weight on high fat diet on room temperature, and compared to control mice, these mice gain more weight as 2 to 3 weeks later after high fat diet. And then just so somewhat this is expected because we expect that the brown beige fat loss leads to reduced energy expenditure, and eventually they gain more weight. However, though, what we didn't expect at the time is that these mice, EHMT knockout mouse, develop severe hepatic steatosis. This is a H&E staining of the liver of the knockout mouse versus control. And these knockout mouse have really accumulate triglycerides in the liver, and we call hepatic steatosis. And also they develop a severe insulin resistance in the liver and the muscle. And then later, Yutaka Hasegawa actually showed that this fat-specific knockout of EHMT1 leads to adipose tissue fibrosis and inflammation. And these phenotypes are unexpected at the time because these phenotypes are very different from UCP knockout mouse. Again, UCP knockout mouse are not diabetic as long as you keep mice in room temperature. And then, on the other hand, brown or beige fat deficiency leads to many metabolic defects such as insulin resistance, hepatic steatosis, adipose tissue fibrosis, etc., which is not seen in UCP knockout mouse. So I want to mention that this EHMT1 knockout mouse really is consistent with the phenotype of the PRM16 knockout mouse. This is the work by Bruce Spiegelman, led by Paul Cohen, 2014, where PRM16 was knocked out only in the fat cells. And again, this is a body weight on a high fat diet on room temperature, and knockout mice gain more weight, gradually more weight as compared to control mice. And then, but what is really the key data here is that a hyperinsulinic euglycemic clamp show that the mice, knockout mouse, develop severe insulin resistance. And this occurred way before this is a clamp time point. And so the insulin resistance occurred much earlier than the difference in body weight emerges. Again, so these really data, PRM16 knockout or EHMT1 knockout mouse studies suggest that, or really these were convinced us that there must be something more than UCP1 in brown and beige fat. So the question is, how can we identify such UCP independent mechanisms? So to this end, Kenji Ikeda in my lab, now he's assistant professor in Tokyo, he genetically activated beige fat biogenesis mice that lack UCP1. So here's a transgenic mice expressing PRM16 only in the fat cells, but lack UCP1. And when we put these mice in cold temperature, as you see, this y-axis is the core body temperature of the mice, UCP knockout control versus the UCP knockout, yet have higher amounts of beige fat. And then UCP knockout mouse quickly developed hypothermia. Core body temperature went down below 30 degrees, close to 25 degrees, and really sort of hypothermic phenotype. However, when you activate beige fat biogenesis, and this hypothermic phenotype was completely rescued. And then, of course, core body temperature can be influenced by many factors, for example, insulation or shivering, and et cetera, et cetera. So we directly measure the tissue temperature. Here, y-axis is a temperature difference, delta temperature of the mice. And then when we add norepinephrine, this mice that express PRGM16 means that have more beige fat, but lack UCP1. The tissue temperature went up as soon as you inject norepinephrine. So this really provided direct evidence that a beige fat can make heat, even in absence of UCP1. What is really surprising is that when you put those mice on high-fat diet, again, it's a room temperature, y-axis is the body weight, these mice with more beige fat gain less weight as compared to control UCP1 knockout mouse. But just suggesting that anti-obesity effect of beige fat is UCP independent. And what is also shocking to us is that if you measure glucose tolerance test, or insulin tolerance by GTT or ITT, you see that the mice with more beige fat are more insulin glucose tolerant and insulin sensitive compared to control mice. Again, suggesting to us that anti-diabetic action of beige fat is also UCP1 independent. Again, these data are really shocking to us, and we really want to understand the molecular mechanisms. Okay, so really these works transform the way that the role of brown and beige fat goes far beyond UCP1 thermogenesis. And this is certainly involved many mechanisms, but I want to mention a few things. For example, clearly thermogenesis is an important aspect of brown fat or beige fat. And then really for the longest time, it's all about thermogenesis by UCP1, but clearly there's UCP independent pathway exists. For example, I'm going to briefly overview calcium cycling that we identified. Brice Spiegelman's lab showed that creatine cycling is involved, and Mitch Lazar's lab over 10 years ago showed that triglyceride fatty acid cycling exists in there. So there are multiple pathways to make heat independent of UCP1. Also, another important yet UCP independent mechanism that regulates, particularly involving insulin sensitivity of adipose tissue, is that a beige fat is associated with angiogenesis, and also repressing adipose tissue fibrosis and inflammation. And these factors are again UCP independent. And then I don't talk much about this, but the many labs from independent groups show that brown and beige fat secrete a variety of molecules, peptides, lipids, or microRNAs, and we call cobatokines or adipokines arise from brown adipocytes, and it's cobatokines. And these are appear to be important for tissue or systemic level, particularly glucose homeostasis or lipid homeostasis. Then the last part is the metabolic sink. We know from the study of FDG PET scan that brown and beige fat are a sink for glucose, but also clearly brown beige fat actively uptake fatty acids. And I'm going to briefly later talk about branched chain amino acid. But again, these sort of those sort of mechanisms are coordinately regulating systemic glucose or lipid homeostasis, and these are not necessarily UCP independent. Okay, so particular interest for us regarding the UCP independent mechanism is the calcium fetal cycling beige fat, where we found that in Kenji's work show that this beige adipocytes have unique calcium fetal cycling that involves SARCA2b and ryanodine receptor 2 and IP3 receptor, where they actually, SARCA2 uncouples this calcium import from ATP hydrolysis in response to norepinephrine. And then this is molecular components are different from skeletal muscle, but we know from the literatures that the mutation in ryanodine receptor 1 cause malignant hyperthermia. And then this is called SARCA1 and then ryanodine receptor 1. On the other hand, beta adipocytes use SARCA2b and ryanodine receptor 2 and IP3 receptor. Cardiomyocytes actually use SARCA2a and ryanodine receptor 2. So, and this skeletal muscle malignant hyperthermia is more about dysregulated, uncontrolled calcium fetal cycles. However, beta adipocytes use SARCA2b. This is more regulated, norepinephrine stimulated calcium cycling. Mechanistically, I should mention that this is very exciting, particularly from evolution perspective. I told you earlier, pigs don't have UCP1, but it turns out that pigs actually use calcium cycling to make heat. So here's one of the examples where we isolated pig adipocytes from subcutaneous adipose tissue. And then when you add norepinephrine, then upon norepinephrine stimulus, the adipocytes from the pig adipocytes start to make heat based on this thermal probe. And then when we knock out or knock down SARCA2 by shRNA, this oxygen consumption rate, y-axis, is that the respiration of the pig adipocytes are really reduced when you knock out SARCA. So the norepinephrine-induced respiration in pig adipocytes depends on SARCA2. So mechanistically, how does calcium cycling thermogenesis work? Well, it's being actually shown by beautifully shown by Dr. DeMellis and others over 20 years ago, particularly in the skeletal muscle, where SARCA import calcium into the SR or ER against a steep concentration gradient in a 2-to-1 stoichiometry of calcium ATP. Okay, so this is occurs in normally this direction. However, under certain conditions, such as a high ATP-ADP ratio or a reduced binding affinity of SARCA2 to calcium, then calcium, the SARCA2 can be exothermic. And then although this estimate of how much heat generated from calcium uncoupling varies, depends on tissue. So this is just based on the skeletal muscle. But in the skeletal muscle, how this calcium binding affinity is regulated is mostly done by the micropeptides like phospholamban or myoregulin. These are transmembrane micropeptide that binds to the SARCA1, and this is sort of the key regulator of this calcium mediated thermogenesis, particularly the skeletal muscle. So we've been really searching for this, but so far, none of the known micropeptide exist in beta adipocytes. So we must be something, the micropeptide exists that binds to SARCA2b and then regulates ATP hydrolysis activity that may lead to thermogenesis, but we haven't discovered yet. So we're actively searching for it, and I hope to update you sometime soon. But regardless, though, this discovery of calcium fetal cycling beta adipocytes really, it makes us think about how to intervene this pathway. And one of the approaches is to selectively activate calcium cycling only in fat cells by using optogenetics. And in this, if this pathway is active enough, then we should be able to stimulate this calcium fetal cycling by optogenetics without cold exposure. And then we call this idea called fat-specific co-mimetics. And this is important because cold exposure or a beta-adrenergic receptor signaling increase blood pressure. And this can be detrimental to the cardiovascular disease. So that Therefore, if we can bypass that cold exposure per norepinephrine signal by directly activating calcium cycling in fat cells, maybe we should be able to improve metabolic health without cold exposure. So this idea of the selective fat-specific co-mimetics became possible when we team up with Dr. Ada Poon at Stanford. She's an engineer scientist who developed this wireless optogenetic technology, and where her lab actually developed really cool implantable wireless optogenetic device where we can implant directly into the subcutaneous adipose tissue of mice. And you can see here in sort of free living mice with the optogenetic device, that we can actually selectively activate this calcium flux only in the fat cells in the subcutaneous adipose tissue. So just a note, in this experiment, we overexpress channel rhodopsin 2 only in the fat cells by using adiponectin Cree. And then a light pulse stimulated the intracellular calcium levels, showing this y-axis. And this is through an L-type channel. So here, when we stimulate calcium cycling on by light, and then when you treat the cells with L-type inhibitor, L-type calcium channel inhibitor, then you see that this light-induced calcium flux is significantly blocked, suggesting that L-type channels require for that light-activated calcium flux. And then also, we blocked randin receptor 2 as well as IP3 receptor. Then the light-activated calcium surge was completely blocked, again suggesting that a BH5 used both randin receptor and IP3 receptor for calcium cycling, which is consistent with our previous works. Now, the question is really the light pulse sufficient enough to stimulate thermogenesis in vivo? And here's the experiment. So we again implanted this the wireless optogenetic device into the subcutaneous adipose tissue of mice that express channel rhodopsin 2, or control mice which did not express channel rhodopsin 2. And then here's a temperature change, y-axis. And when we turn on the light for up to 10 minutes, which is a sort of cycle, 5 milliseconds on, 95 milliseconds off, so the light, really acute light pulse. And within a few minutes, we found that the subcutaneous adipose tissue started to generate heat, and temperature went up to 0.5 degree. On the other hand, this increase was not seen in control mice, even though control mice also received the same light pulse, but simply they don't express channel rhodopsin 2, suggesting to us that this increase in temperature in the subcutaneous adipose tissue is due to thermogenesis by the light. And then this is really exciting data is that we put these mice on thermoneutral condition, 30 degree, and that means that there's no cold stress, essentially living in Hawaii. And then we measure whole body energy expenditure called VO2. And then y-axis is a whole body energy expenditure using clams apparatus. And then upon light pulse, up to 10 minutes of light pulse, now we see that whole body energy expenditure went up to 35%. And then this 35% of increase in energy expenditure is pretty significant, highly significant. Of course, we can't directly translate this number to humans, but 35% increase in whole body energy expenditure is close to maybe 700 kilocalorie in humans. That means probably this is sort of worth equivalent to 1 hour running. So now in mice, without a cold exposure, 10 minutes light can achieve 35% of energy expenditure without a cold exposure or any exercise. That to us is a very striking number. And now we, Kenji and Kazu Tajima, put these mice on high-fat diet. Here's an experimental scheme where we put implant this device, wireless optogenetics, and then put them on a high-fat diet, and every day these mice will receive light, 10 minutes light, and then you can see this body weight y-axis, and then mice that express channel rhodopsin 2 and activate calcium fetal cycling gain much significantly less weight as compared to control mice, suggesting that the activation of calcium cycling thermogenesis was sufficient enough to reduce body weight gain. And this difference in body weight was exclusively come from difference in subcutaneous adipose tissue and somewhat epidermal adipose tissue. No difference in the liver or muscle or brown adipose tissue in the intrascapular regions. Again, we are very excited about the potency and robustness of this effect, where the calcium, really the fat-specific activation of calcium cycling thermogenesis only for 10 minutes is sufficient enough to increase energy expenditure and also reduce body weight gain. Okay, so back to this. So let me review what we talked about here. So initially we talked about, we studied brown beige for biogenesis, and we know that this activation, either brown or beige for biogenesis, improve metabolic health. And we thought about, really the dogma in the field was UCP and dependent thermogenesis. But surprisingly, activation of brown or beige fat improves metabolic health in a UCP independent manner, and which led us to identify this fetal cycling, calcium fetal cycling, or creatine cycling. However, though, this is not the whole story. Really, this fetal cycling cannot entirely explain everything about brown or beige fat in terms of the improved metabolic health, because the deletion of this pathway was not necessarily sufficient enough to cause insulin resistance in mice. And this is unless mice develop obesity. For example, a blockade of creatine cycling, eventually mice became obese, and then they develop insulin resistance. But the fact is that when we deplete brown or beige fat, but genetically by knocking out EHMT1 or PRM16, mice instantly develop insulin resistance and hepatic steatosis way before we see difference in body weight. So something is really missing here. And this is one of the ongoing questions: why or how brown beige erythrocytes improve insulin sensitivity? And then one of the hints, though, came from the transcriptional analysis of mice that I told you, the PRM16 transgenic mouse in UCP and knockout background, where they have more beige fat but lack UCP1. And we found that these mice have really high upregulated branched chain amino acid or catabolic pathways. And this is one of the most robust pathway that links to improved insulin sensitivity. And then this is particularly interesting and caught our attention, really because many epidemiological studies from independent groups, for example, one from Chris Newgar's lab or Robert Gersten's lab, BIJMC, and many others, show that the circulating BCA levels and also reduced BCA catabolism or oxidation in peripheral tissues are linked to type 2 diabetes. For example, this is one of the signature data from Chris Newgar's work that BCA-related metabolic signature correlate with insulin resistance in human cohorts. I believe this is from Framingham cohort. And so that the idea of brown or beige fat being a catabolic organ and then clear BCAA from the circulation makes sense and then very interesting. So we wanted to follow up on this observation in human subjects with or without active brown adipose tissue. So this is a collaboration with Dr. Saito and Dr. Soga in Japan, where they recruited healthy human subjects with high or active brown adipose tissue or people who did not have active brown adipose tissue. And then these subjects were we received FDG PET scan and before and after cold for 19 degree Celsius for 2 hours. And then we ran serum metabolomics. And in this metabolomic study, unexpectedly found that the circulating level of valine and leucine with branchy-chain amino acid, they are really significantly reduced after cold exposure, but this reduction was only seen in subjects who have active brown adipose tissue. This reduction was not seen in subjects who did not have active brown adipose tissue, implying that the brown beige fat actively uptake BCA from the circulation. And eventually we confirmed this or validate this observation in mice. We used some of the mouse genetic and to validate the brown and beige fat actively uptake BCA from the circulation and oxidize them. And this is also shown by or validated by other groups. For example, Zolrani's lab showed that this is also occurring in brown and beige fat. So then we thought maybe, so what BCA is doing in brown fat. So one of the experiment we did here, this is led by Takeshi Yoneshiro. Now he's a assistant professor in Tokyo. He knocked out BCKDH, one of the rate-limiting catabolic enzyme, oxidation enzyme, specifically in brown adipose tissue. So we use UCP and knockout UCP and CRE to knock out BCKDH. And this is a Western blot validating that BCKDH is knockout in deleted in knockout mouse, but not in controls. And then these BCKDH knockout mouse quickly develop hypothermia after cold exposure, which is consistent with the idea that BCKDH mediated BCA oxidation is required for thermogenesis in brown fat. When we put these mice on a high-fat diet, on again this room temperature, and then in knockout mice that lack BCKDH only in the brown fat, gain more weight as opposed to relative to control mice. And also when we measure insulin tolerance by insulin tolerance test, then this knockout mice, brown fat specifically knockout BCKDH was sufficient enough to cause insulin resistance, which is again consistent with the notion of that brown beige fat are protective against insulin resistance. Then the question is how BCAAs are used in brown fat. Branchy-chain amino acid, let me remind you that is essential amino acid, means that we cannot synthesize BCA in our body. Only way to take this is from food. And we eat proteins and digest them. This BCA, branchy-chain amino acid, valine, leucine, isoleucine. These are critical components of protein synthesis. First of all, needed for protein synthesis, particularly for organs like pancreas that have to make a lot of proteins, particularly insulin. They take a lot of BCAAs and they make proteins. But also this is important, particularly leucine activates mTOR pathway, the nutritional sensing pathway. So the BCAA is really important for the protein synthesis. However, BCAAs are also actively uptaken into mitochondria and then oxidize them as a fuel. And then this is occurred through a mitochondrial localized enzyme called BCAT2, which is deaminase. The question is how BCA get into the mitochondria, because the oxidation enzyme exists in the mitochondria. That means that BCA has to get into the mitochondria through penetrating mitochondrial inner membrane. But we know that in mitochondrial inner membrane is very impermeable to small molecules and ions. So that means that there must be something here we call carrier proteins that mediates BCA transport into the mitochondria. And so we really decided, this turned out to be a very important and long-standing question in the field, and we really wanted to hunt for this unknown mitochondrial carrier for branchy-chain amino acid. However, though, it turns out to be very challenging because mitochondrial flux is very rapid to capture by existing sort of conventional approaches like mitochondrial purification. It takes about 90 minutes to do that, but mitochondrial flux is so rapid that we need another way to measure this. And also the another technical challenge is that reconstitution of mitochondrial carriers, where often mitochondrial carriers in the inner membrane need multi-subunits, and so and then has to be in the right orientation to be functional. So the reconstitution system is also very challenging to develop. Therefore, the field of this mitochondrial metabolic carriers are sort of it's being very challenging for many years. So therefore, we applied new technologies. For example, really the tools that developed by David Sabatini's lab in MIT use this mito-tag mice, where the outer membrane mitochondria is GFP tagged, so that we can rapidly purify active mitochondria within 12 minutes in vivo, and we can measure flux in the mitochondria. We call mitochondrial metabolomics. And then this can be done in many other tissues. We do brown fat, beige fat, muscle, liver, and other tissues. And then in terms of the reconstitution, we can actually isolate large amounts of native inner membrane from brown adipocytes. Again, I told you that the brown fat contains mitochondria at the highest density, so that we can obtain native inner membranes which contain high cardiolipin, and then reconstitute them into the mitochondrial liposome together with the recombinant proteins made from insect cells. So we call mitoliposomes, so that way we can really reconstitute mitochondrial carriers in the right orientation in a known cell-free system. It's called mitoliposome. And really these new tools transformed our approach to study mitochondrial carriers. And with these tools, we identified new, previously unknown membrane protein called SLC2544 as the first mitochondrial BCA carrier, and we call them MBC. And this is, first of all, this is this data shown from isolated mitochondria, where wild type control cell versus SLC25A44 knockout cells. We purify mitochondria from those cells, and we measure the flux or import of these indicated metabolites, and we found that only valine leucine import was affected in the absence of SLC25A44. Other amino acids appear to be fine. And then another experiment is mitoliposome. Particularly, this is spearheaded by Takeshi, Yuneshiro, and then Chen Wan. They generated recombinant SLC25A44 and reconstituting the mitoliposomes. And then here is the uptake of leucine, show that SLC25A44 including mitoliposome actively uptake leucine. So this is sort of the reconstitution experiment where this molecule mediates a leucine import into the mitochondrial membrane. And then, so in vivo function here, Takeshi and Chen show that when we measure tissue temperature of brown adipose tissue and muscle of control mice, as well as the bat-specific knockout for MBC, and if you inject norepinephrine, you see that control mice can generate heat. The temperature of the tissue, temperature of brown adipose tissue rapidly increased within a few minutes, but this induction was significantly blunted or reduced in SLC25A knockout mouse. No difference in skeletal muscle in response to norepinephrine. And then when we measure BCA oxidation in the mitochondria, then this MBC knockout must have significantly reduced BCA oxidation in the mitochondria. And then instead, this oxide, since BCA cannot get into the mitochondria of knockout mouse, so eventually circulating BCA increased in knockout mouse. So this is plasma BCA levels, y-axis, is significantly increased in knockout mice. And eventually, Takeshi actually found that the protein synthesis is also enhanced in knockout cells. What is really exciting and then also really validate our observation here is that glucose tolerance test and an insulin tolerance test showed that the whole body knockdown of the MBC, mitoBCA carrier, was sufficient enough to cause insulin resistance and glucose intolerance. GTT shows that the knockout mice have impaired glucose tolerance and for insulin sensitivity as well. And if you look at the tissue level insulin signaling based on the phosphorylation of AKT, then you can see this in response to insulin injection plus the insulin, and the insulin injection rapidly increased phosphorylation of AKT in brown adipose tissue, but this injection was significantly blunted in knockout mice. Again, showing that the MBC is required for systemic glucose homeostasis and insulin sensitivity. Okay, so here's our journey. We started with the fundamental understanding of brown and the beige adipocyte development, and then through understanding transcriptional factors or progenitor cells, now we're able to manipulate, either activate or block brown beige fat development. So we can really, this allow us to study the biological significance or biological role of brown beige fat in vivo. Then what we found was that activation of brown beige fat improved metabolic health, such as improved lipid homeostasis, improved glucose tolerance, improved insulin sensitivity in mice, and later many studies from other groups show that this is the case for humans as well. The long-standing dogma in the field of brown fat was all about thermogenesis by uncoupling protein 1, and this really the metabolic health maybe links to UCP-mediated thermogenesis, but surprisingly, we found that the metabolic benefit of brown or particularly beige fat is nothing to do with UCP1, UCP independent. And this led us to identify UCP independent mechanisms, including calcium fetal cycling. And then really the important aspect, particularly in the case of improved insulin sensitivity, is about clearance or disposal of branched chain amino acid. And this occurs through mitochondrial BCA carrier that we identified. So with that, I'm sure there are other aspects of brown and beige fat that links to improved metabolic health. Again, I told you earlier that the tissue homeostasis is an important part, and the release of bradykinin could be another aspect of it. So there are many new things are coming up of this field, and I really believe that brown beige fat has sort of a discovery tool to understand how bioenergetics is in brown beige fat is important for systemic regulation of energy homeostasis, glucose homeostasis, and insulin sensitivity. With that, I'm going to end my talk by thanking my lab members. Particularly, we were University of California, San Francisco, and we moved to Boston, joined the Harvard last year, and Chen Wan actually came with me from UCSF, together with Chen Wan and Takeshi. Now he's a faculty in University of Tokyo. They led the work on the identification of mitochondrial BCA carrier. Kenji Ikeda is a faculty, and also the Kazu Tajima, they really led the work on calcium cycling thermogenesis with the collaboration with Ada Poon's lab. I should say, I think Dr. Saito and Soga in Japan, they really closely work with us on the human metabolomics, and also other collaborators. And of course, I want to thank funding sources, NIDDK, and another fundings. Thank you very much for your attention. And if you have any questions, feel free to contact me. And then I will leave the conclusions point here to end my talk. Thanks again.